Okay, first I'd like to say a thank you to Cytel for inviting me to give this talk and for their hospitality whilst I'm here. That's gratefully received. In the same way that Robert was setting expectations, it's probably worth saying a little before I start this talk in that it's somewhat exploratory. It doesn't come up with a nice, neat solution. So I think its role is really to make you think about this topic because I believe it's somewhat neglected. We had a number of discussions when I was at Pfizer about it without ever, I think, getting particularly close to resolving what we might do about it. Pfizer's a big company, of course, so I presume that other companies, other people have had similar thoughts. But it would be good to provoke some thinking about this and see if this potential problem is something that we should think more about and do more about than perhaps we currently do. Okay, so as you can see, the title for the talk is Selection Bias for Treatments with Positive Phase 2 Results. So I'll expand on what I mean by that by way largely of example in a minute. I'd just like to acknowledge before I start the role of Christy Chung-Stein and David Lee. So Christy Chung-Stein was head of the Statistical Research and Consulting Group at Pfizer for quite a number of years. She left in 2015 and, as you can see, set up her own consultancy, working largely in the US, but also Europe, I gather. And David Lee is a colleague who's still at Pfizer. So we were all Pfizer at one time, but only one of us is now, and that's David. Okay, so what's the sort of structure of the talk? So in terms of content, I'd like to talk a little bit about what I mean by selection bias problem. The work that's contained in these slides was submitted to Pharmaceutical Statistics. We've had the usual sort of response that says, yes, it's of interest, but could you change this, that, and the other, which we will attempt to do. But interestingly, when we first submitted elsewhere, one of the referee's comments was, even after reading the paper, was, I don't understand what the selection bias problem is. So hopefully we'll try and clarify here, and hopefully people will understand what the potential problem is. We'll then talk about some previously proposed corrections for the bias arising from the selection bias. Talk about two new proposed corrections for bias, new at least in this setting. Then need to talk about an informative prior for one of the previously proposed approaches, which is essentially just a Bayesian analysis. We'll look at some measures of performance, then we'll look at some results and some concluding remarks. Okay, so I think it's perhaps easiest to rather than try and talk about this theoretically, to look at some simple numerical examples, because then you start to get an appreciation perhaps of what's going on numerically at least. Okay, so the first example, so we're talking about the simplest possible situation here. So we're going to talk about a parallel group trial with two treatments comparing a new treatment with a control. We're going to say that the sample size is 50 subjects per group. Going to denote the endpoint of interest by X, the mean for the treatment group, mu T, the mean for the control group, mu C, and we're going to assume that the variance is known and equal to 1, and that higher values indicate a better result. So we're trying to think of a really simple situation to illustrate what we're about, and we're going to have a simple Z statistic, and we're going to declare the trial a success if our Z statistic is greater than the 0.95 or the 95% point, which if you did that little bit of calculation would say that your observed treatment difference has got to be greater than 0.33. So that will be one example. As I say, we're trying to keep things just about as simple as possible. And the second example only differs in that, so instead of saying that a treatment difference has to be greater than 0.33, we're going to say treatment difference actually has to be greater than 0.5 before we're interested in the trial result. Okay, so two fairly simple situations there. So what we can do is we can use to delta TOR, or to to denote a generic estimate of the treatment difference. And we're going to consider a selection rule which says we're going to select a treatment if actually the treatment difference is greater than some predefined difference that we wish to have. Previously we spoke about 0.33 and 0.5. So delta PD denotes the threshold for a positive decision. So if we have any generic estimate, delta TOR, the bias is going to be given by the expectation of that, but it's conditional because we're saying we have to have a positive result given by delta greater than delta PD. So it's that expectation with that conditional element minus delta that's going to give us our bias for any estimator delta TOR.
That's the decision rule for that treatment going forward.
Yes.
And that decision rule doesn't depend on the standard error. It's just that the point that's what the company expects to see regardless of standard error, is it?
Well, it could implicitly, like that first example was implicitly dependent on the standard error because it was a Z statistic. But the second example was just a number, just a straight cutoff.
A straight cutoff
for the second example.
Just the estimate.
Yes, yes. So this is all talking in terms of our usual estimate to make a decision to go forward.
And that's, in industry, that's a well-known way of making decisions regardless?
Well, certainly advise, well, it could be. I mean, obviously in practice, decisions could be more complex, but if you want to reduce it to something that's a rule, then people have in the past made decisions based on said statistics. Certainly at Pfizer, we've made decisions based on target values, as we call them. So the 0.5 would be an example of a target value that we would have to surpass to be interested to take a drug forward. So, yeah, things that have been used. Okay, so we can evaluate the bias of any estimator we want to use conditional on the fact that we've got a result that we're interested in by needing to pass some threshold. So actually, and sorry, I should have said, and if we put our usual estimator in, so we've got the usual estimator and the usual estimator has to exceed the threshold, then you can just go ahead and evaluate that bias for the usual estimator plotted against the true treatment effect. So on the horizontal axis, we've got the true treatment effect. On the left-hand side, we've got example 1, right-hand side, example 2, and we've got bias on the vertical axis. So the fact you can see that only looking at results that surpass a decision threshold, whatever the value of the true treatment effect, is going to lead potential for bias, at least certainly for low values of the true treatment effect. Only when you get to higher values of the true treatment effect does the bias that comes about from having a decision threshold actually go to 0. And you can see that the biases, you know, you might consider them fairly large, so not insignificant in this particular example. A plausible question is, well, okay, I picked a sample size of 50. What happens if I look at bigger sample sizes? Maybe you think 50 is quite a small sample size, although it depends on the sort of phase 2 study that you're looking at. And as you increase the sample size in the example 1, you can see the curve moving in towards the 0 point, if you like, the fact that the bias is heading towards 0. And example 2, where we had that the threshold was that you had to exceed the value 0.5, you can see that actually it's rather more difficult to get rid of the bias by just increasing the sample size. Although with all these graphs, to be fair, you have to point out that with a bigger sample size, the probability of passing a threshold with a low treatment effect is actually going to become quite small. So you have to be a little bit careful interpreting this, because if you have a sample size of 1,000, it's unlikely that you're going to pass a threshold with a very low treatment effect. Can happen, the probability isn't 0, but the probability will become quite small. Okay, the other thing you might ask about, particularly in relation to thinking about that second example, is what happens if I increase that decision threshold, or at least I change that decision threshold? So the central black line is that 0.33, so actually corresponding to the first example. As you go upwards, the decision threshold moves to 0.4, 0.5, 0.6. As you move the decision threshold up, you can see that the bias moves upwards. As you move the decision threshold down, the bias comes down with the lower curves, 0.3, 0.2, 0.1. So sample size and threshold can be illustrated to have an effect on the bias when plotted against true treatment effect. Okay, so there has been previous work, although not necessarily directly thinking about selection bias, but in effect that's what they've been doing. So Wang, Hung and O'Neill proposed that the usual treatment estimate should be reduced by 1 standard error when you sample size for phase 3 using observed phase 2 results to guard against basically this selection effect. And they considered cases where a phase 3 trial is launched if you take the observed difference, so you don't make an adjustment, you take the observed difference minus 1 standard error, or the observed difference minus 1.96 standard errors, and you insist when you subtract 1 standard error or 1.96 standard errors that that adjustment has to exceed the launch threshold. So it's not then just a question of the observed value exceeding the launch threshold, the adjusted value has to exceed the launch threshold for you to be interested to go and launch a phase 3 trial. And the upshot of the simulation work they did was that they found subtracting 1 standard error approximately equates planned power, so planned power coming from your sample size estimation, with average power in the phase 3 trials that are actually launched. So there's a big difference potentially between the power that you plan and the power that you actually observe in the trials that qualify to launch because they've exceeded that launch threshold, either using an unadjusted or an adjusted estimate. So we'll refer to this method as the 1 standard error method. So that's Wang, Hung and O'Neill's recommendation from a few years back. We looked at the same scenarios as Wang, Hung and O'Neill looked at, but instead of talking about equating planned and average power for the phase 3 trials launched, we considered equating estimated assurance, which you'd calculate at the end of phase 2, what's the probability of your phase 3 trial being successful, integrated over the prior obtained from your phase 2 results. So equating estimated assurance using estimated parameter values with so-called theoretical assurance, where you substituted in the known theoretical values in the simulation. And the event of interest, of course, is achieving statistical significance in your phase 3 trial. So we considered launching a phase 3 trial. So whereas Wang, Hung and O'Neill took off different multiples of a standard error, we said, well, maybe you could multiply the observed difference by some fraction less than 1 to discount or downweight the observed phase 2 results. And broadly speaking, we found that multiplying by 0.9 did a reasonable job of equating estimated and theoretical assurance, provided the phase 2 trial wasn't too small and the launch threshold wasn't too large. So we'll refer to that approach as the 0.9 multiplication method. So we have the 1 standard error adjustment and the 0.9 multiplication method. So Zhang, in a conference presentation, said, well, he would consider using a Bayesian approach with an informative prior, and then he would take the difference between your usual estimator without using the prior and the estimator using the Bayesian prior, i.e. your posterior result. He would take the difference to represent selection bias. So he would view the Bayesian analysis with an appropriate informative prior as making a suitable adjustment for this selection effect. So Zhang's method is basically a Bayesian analysis with an appropriate informative prior. So we'll refer to that as the posterior mean informative prior method. Okay, so having seen that graph earlier of bias plotted against true treatment effect value, you can start to think of maybe there are other ways you might estimate and correct for bias. So one approach might be to estimate average bias to your usual estimator, using this decision rule based around the usual estimator, by weighting the bias at a specific true treatment effect by the likelihood of the treatment, particular treatment value. So that expression there is all that's all that is doing. It's weighting the bias by the likelihood for that particular value of the treatment value. And that'll give us an average bias, okay, integrated over the true treatment. So your adjusted estimator, if we call that delta squiggled core, is going to be delta hat minus A bias. You take off the average bias to get an estimate, an adjusted estimate, and we can call that bias correction the likelihood weighted bias method. Okay, so that's one thing you might do. A second approach might be to say, well, okay, suppose we had a vague prior for the true treatment effect, and we calculated a posterior distribution for the treatment effect using this vague prior, and then we're going to take, say, the 20th percentile point of that posterior distribution for the true treatment effect and pick up the bias that corresponds to that 20th percentile point of the posterior distribution for the true treatment effect. So what this is trying to do is be a little bit conservative because the bias will be bigger associated with the smaller treatment effect. So this will make a bigger adjustment than the A bias, subtracting average bias. So we could get another correction here, delta minus so-called percentile bias, where it's the 20th percentile, which will give us a more conservative adjustment than using the average bias. So we could refer to this as the percentile bias method. So that's a couple of approaches for something you could do to make alternative corrections. To go on and look at some results, before we do that, for that posterior mean method, for the Zhang's approach, which is basically the Bayesian analysis, obviously you have to decide what sort of informative prior you might use. So for the purpose of obtaining results here, what we've used is a mixture prior where 50% of the mass assigns no effect, no treatment effect whatsoever, and 50% uses an exponential distribution I'll look at in a moment. This is not totally plucked out of the sky in the sense we did take a look at Pfizer's overall phase 2 results over quite a number of years, and you get this sort of picture coming out. There's quite a lot of, well, I suspect it's the same for all companies. There's quite a lot of drugs that do nothing or virtually nothing, and then you get this sort of tail of drugs and a rather few drugs that have a big effect size. And this prior distribution reflects that sort of approach. So it's something that does have some basis in reality for what we've observed for Pfizer's results going back, I can't quite remember how many years, might be 10, 15 or more years. And that would be the exponential part of the mixture prior, so you can see chances of getting a big effect size are rather small. So this is considering, if you like, this is a sort of generic prior for all the drugs that come through development without any further information about a particular drug. You could obviously start to look at particular types of drug, and maybe the distributions would be rather different to looking at the distribution as a whole for all drugs in development. Okay, so we can look at a number of measures of performance for these various bias corrections. We can look at the bias of the bias-corrected estimates. We can look at the mean square error. So delta squiggle core now is any of the estimators. Something else we can do that is likely to be of interest is, given that your usual estimate has crossed a decision threshold, does your bias-corrected estimate also cross that decision threshold? So you would maybe like the sort of situation where maybe your usual estimated crosses the decision threshold. Well, suppose it crosses the decision threshold, possibly for quite low treatment values. In that case, you would like your alternatively corrected, one of your corrected estimators not to cross that decision threshold if the true treatment effect is low. And by comparison, if the true treatment effect is of interest, then you would like it to cross the decision threshold. So we can plot this against the true treatment effect and see how that correction does, given that your usual estimate has crossed a decision threshold. We've already spoken about average power. So if you went on to launch a phase 3 based on the phase 2 result you've got, then average power for a phase 3 trial is the average power in the trials you actually launch. And we're going to talk about launching on the basis of a bias-corrected effect. And then one final thing we haven't talked about is overall power. So suppose after your phase 2 trial, you launch a phase 3 trial, what's the probability of both of them being successful? So that development program of phase 2 followed by phase 3 being successful. And we can refer to that as overall power. Okay, and just to denote in this simulation, or the results rather, phase 3 trial is calculated as usual with a two-sided 5% significance level and 80% power. Okay, for average power and overall power, there was a further paper more recently by DiMartini where he actually comes up with another method, but his concentration is actually on overall power, that idea of having a successful phase 2 followed by a successful phase 3. So he, like Wang Hung and O'Neill, is using the lower limit of a confidence interval, is allowing that to vary to achieve a particular desired overall power. So instead of talking about just the power of the phase 3 result, he wants to achieve an overall power for that development program as a whole, consisting of the phase 2 and the phase 3 trial. Just one thing to note, although he uses this adjusted estimate to come up with a sample size for phase 3, he doesn't use the adjusted estimate to decide to launch a phase 3 trial, which to us seems a little curious, because if you think you need to make an adjustment to get a better estimate of treatment effect, it would seem logical then to say, I need that better estimate to exceed my decision threshold before I launch a phase 3 trial, rather than going back to the uncorrected estimate saying, well, as long as that passes a decision threshold, I'll go to phase 3, but I'll use my corrected estimate to calculate the sample size for that phase 3 trial. And he called his method the calibrated optimal strategy, or COS is a bit less of a mouthful. Okay, so you can start to look at what these corrections look like, and hopefully we can pick out in the graphs the various ones. So 1SE, which is at the top of the legend, is the one hang on an eel. That's the dark blue line, which is the constant correction as you move along the x-axis, which is observed treatment effect. Y-axis is going to have the adjusted treatment effect on it. So the one standard error adjustment is that constant adjustment as you move along the observed treatment effect. The 0.9 multiplication starts close to the 45-degree line and gets further away as the observed effect gets bigger. The posterior mean informative prior heads towards the 45-degree line but doesn't ever intersect with it. The likelihood weighted bias approach, which is the blue line, eventually coincides with the 45-degree line as the observed treatment effect gets bigger. And the percentile bias, the brown line, which is the biggest adjustment for low observed treatment effects, eventually heads up and again intersects the 45-degree line. So that's probably clearer to see in example 1 than example 2. So there's different sorts of corrections going on there when you look at observed treatment effect and what the adjusted treatment effect is corresponding to an observed effect. Okay, so we can start to look at some of the comparisons, look at some of the measures to compare the methods. So we can look at bias, compare the bias of these various corrected results. So again, 1SE is the dark blue line, which comes down and eventually, you could say, overcompensates for the bias as the true treatment effect gets bigger. We've now got true treatment effect on the x-axis. 0.9 multiplication starts with quite a high bias, comes down, again, sort of overcorrection. Posterior mean for the informative prior we're using, low bias initially, but then overcorrection. Likelihood weighted bias, the blue line somewhere in the middle, overcorrection, but eventually it's going to come back to zero bias. And percentile bias starts with quite a severe correction, overcorrection, but again heading back towards zero bias. So you can see things aren't straightforward for low true treatment effects. Things like posterior mean, percentile bias do well, bigger true treatment effects, things like likelihood weighted, even the observed difference do better. Okay, so you could look at the mean square error as another way of observing the different results. And again, you can see there's a mixture of what's going on. Initially, the mean square error is quite low for one standard error, percentile bias, and the informative prior approaches. The mean square error then goes up for something like one standard error approach. It's a true treatment effect becomes large. Informative prior is quite large at the end, the mean square error. And the others are sort of converging to something more similar. Okay, something else we talked about is what's the probability the corrective effect is going to exceed the decision threshold, given that our observed effect exceeds the decision threshold. So on the left, we might be interested to say if we took that threshold of 0.33, below 0.33, we don't want to progress. Above 0.33, maybe we don't mind progressing to a further stage of development. And you can see things like the 0.9 multiplication method tends to progress, has high probability of progressing, even if the true treatment effect is small. The other methods are somewhat lower. The weighted likelihood is somewhere in the middle. But obviously, as soon as you move above 0.33, you would like to progress based on your decision threshold. So then you would like to have a high probability of going on to progress to phase 3. Example 2 is there you'd probably like to progress if you get a true treatment effect greater than 0.5. So you'd like a method to have a high probability of progressing above 0.5, but not below 0.5. Okay, and in terms of average power, so this is if you then use the corrected effect to go on to launch a phase 3 trial, using that corrected effect to sample size that phase 3 trial. And what you can see is the dark blue is the one standard error approach. That does well in a sense. The informative prior approach also does well, and the 0.9 method actually doesn't do too badly. But you sort of pay for the good performance of these methods by the fact that if the true treatment effect is larger, you're ending up overpowered. So again, things are not totally clear-cut in terms of what might be the best thing to do. And then we can look at overall power, so that's the combined probability of the phase 2 and the phase 3, the whole development program being successful. And bizarrely, you now come back to your observed difference, just using your observed difference to make a decision as being possibly a not such bad thing to do. The bad thing about it may be the fact that overall power is not a great measure and that you're going to launch potentially a lot of trials on the basis of if you just use your observed treatment difference, which is going to be a very expensive approach to development, launching lots of phase 3 trials that will fail. So again, it's sort of it's open to interpretation, but I think our view is that overall power is maybe not something that we would focus on. Maybe average power would be a better thing to focus on, average power in the phase 3 results. Just to reassure you that we looked at a number of other scenarios. So those were all based on those 2 initial examples, sample size 50. We looked at bigger sample sizes. We looked at different thresholds. Although there are some minor sort of almost aberrations, broadly speaking, the relative performance of the methods was similar to the 2 scenarios outlined here. Okay, so by way of some conclusion, I think we have to face the fact that selection bias can be an issue. I mean, Pereira, in a fairly influential paper, sort of picked this up a little while back, saying he'd done an examination and most large treatment effects he found emerged from small studies, and when you did the additional trials, the effect sizes typically became much smaller. And there's more than selection bias involved in that, but it does lead you to believe that selection bias is also part of that. This is rather stark in the sense of assuming that you have a single phase 2 trial, you have a decision threshold you're going to use, and you're going to make a decision. Certainly at Pfizer, although we were veering towards, you know, as people put more and more pressure on you to accelerate to phase 3, there was more pressure to focus on single phase 2 results. Our previous approach to development had always been to look for similar results for other similar compounds in our portfolio, or to look externally for results, or to do phase 2b trials. So in those sorts of situations, you can have some confidence that you're diluting this selection effect when you combine the results with results from elsewhere and other considerations. I think the danger possibly is that the pressure becomes so much, you conduct one phase 2 trial, you have some sort of strict decision rule, and you progress, and then this sort of thing, I think, probably can manifest itself problems with selection bias. So what would our recommendations be? As I said, I tried to say at the start of the talk, this is somewhat exploratory. We're sort of raising this potential issue. I think it would be good for people to think about it. Our view is it would be good to plot that bias function against the true treatment to see if you're using a decision rule and a particular sample size, what that bias looks like, to be aware, you know, if it is potentially a big thing. Very tentative conclusions from this would be, if you're in the sort of situation here, that maybe the likelihood weighted method is a way of evaluating results generally to think about further development, because it kind of has a middle way between not adjusting and some of the more severe adjustments. On the other hand, if you're going to go into phase 3 immediately and you're concerned about power, then for average power, that 1:c method and the posterior mean for the particular prior we used here did well in keeping your power up for more moderate treatment values. It overpowers for big treatment effects. And I'm really not sure about overall power, so although uncorrected comes out best for that, I think the price may be too high to pay in terms of development costs to really go ahead with that. I think just a single trial with using an observer result might be a somewhat dangerous way to go. And with all of this, because you've seen there are differing results across the methods as the true treatment value varies, it does possibly cry out for integrating some sort of integrated approach over possible true treatment values for a measure that you think is particularly of interest to you to come up with a method to use. Okay, so I think that's about it. Those are the references I referred to during the talk. Okay, thank you.